Welcome to Massoudi Foundation podcast. Today we have very interesting topic with our great dear Dr. Massoudi. The topic is, as you know, CRISPR is gene editing, is a genetic engineering technique in molecular biology by which the genome of living organism may be modified. It is based on simplified version of the bacterial CRISPR-Cas9 antiviral defense system, and by delivering the Cas9 nuclease complex with a synthetic guide RNA into a cell, the cell genome can be cut at a desired location and also allowing existing genes to be removed and or new ones added as well. The technique is considered highly significant in biotechnology and medicine, so we are going to talk about it today with our great dear Dr. Massoudi. Hello, dear doctor, how are you doing?
I'm doing quite well. How are you doing, Dr. Assefi?
I'm great, thank you so much. Dr. Massoudi, one of the questions that our audience has sent us is, what is the role of our immune cells in our fight against cancer?
I'm so excited to have this conversation with you, Dr. Assefi. What we're talking about today is of paramount importance in our ability to fight infections, treat rare genetic disorders, and equally importantly, give us another effective tool in our fight against cancer and cancer cells.
Yes.
The immune system in our body is designed to detect anything and all things foreign and attack and destroy these foreign cells. And of course, tumor cells, by the virtue of the fact that they've undergone mutations and genetic alterations and they're growing abnormally, they are foreign cells to the body, and modifying the immune system in a way that it can specifically recognize these tumor cells and fight them and destroy them is a very effective and attractive proposition in our ongoing fight against cancer. You, at the beginning of the podcast, mentioned CRISPR technology, and the development of this platform has been very exciting in the realm of fight against infections, treating genetic disorders and cancer. CRISPR literally stands for clustered, regularly interspaced, short palindromic repeats, and these are the areas of the DNA that are susceptible to changes, alterations, deletions, and replacements. And essentially, the CRISPR technology allows the guidance of nucleases, which are enzymes that can really remove and alter base pairs in the genome. So CRISPR has become a very exciting and effective genomic editing mechanism. And as such, it has great potential. The two folks that developed this technology won the Nobel Chemistry Prize in 2020 because of its profound effect on the future of scientific breakthroughs.
Oh, great. And what are the two general immune responses in the body?
When it comes to infection or really attacking any viruses, bacteria, or cells that are foreign to us, we either mount a cellular response through T cells and killer cells, or we mount an antibody response through our B lymphocytes that generally elaborate antibodies specific for a particular bacterial infection. So the modulation and augmentation of the immune system in the fight against infections and cancer really has to do with alteration of the mechanism of action and the behavior of T cells as well as B cells that elaborate antibodies.
Yeah, sure. And would you please describe a little bit more about the CRISPR technology?
When it comes to the fight against cancer, there has been really exciting recent developments, and CRISPR technology has been used to take the T cells, T lymphocytes, and genetically alter them to recognize cancer cells. Of course, once the T cells themselves become genetically modified, they could be subject to the attack by other T cells, recognizing them as foreign. The new technology has really succeeded in making the genetically altered T cells undetectable by our regular immune cells. These genetically altered T cells that are altered through the CRISPR technology and the application of endonuclease enzymes can then recognize tumor cells, really essentially poke holes in the membranes of these cells and get into the cytoplasm and the nucleus and start degrading the DNA of the tumor cells and destroying them. The most recent exciting application of this is in the treatment of non-Hodgkin's lymphoma, where a few patients who had become refractory to regular chemotherapy and other treatments underwent the development of the tailored T cells through the CRISPR technology, and they were treated with this, and they are now tumor-free, and their refractory non-Hodgkin's lymphoma has so far been controlled and treated and cured, which is a very exciting breakthrough in our ability to treat cancer cells and cancer in general. And of course, non-Hodgkin's lymphoma is a starting point. I envision a future where the application of this technology can really be a very effective tool in treating brain cancer, breast cancer, and other solid organ tumors as well.
Wow, great news. And what is the role of genetic editing in control or cutting cancer?
Yeah, I think the role of genetic editing, the alterations of the genetic sequence of our fighting T cells or our cancerous cells is immense, and the potential for it is really limitless and boundless. CRISPR technology can be directly applied to cancer cells and essentially delete base pairs that would render them helpless, and they at that point would not be able to replicate and die. Or it can, as we have been talking about during this podcast, really genetically edit our own immune cells to really become the commandos in disguise behind the front lines and go after the tumor cells and kill them while escaping our normal immunological surveillance.
Wow, that's great. That's awesome. Thank you so much. It was a great program and a very interesting topic. Thank you so much, dear doctor.
Thank you, Dr. Assefi. Have a great evening.
You too. Please stay with us. Don't go anywhere. We have so many interesting programs and we have interview with experts. So please stay with us.